David Chung and Max Wynn at UT Southwestern, who made BT2, this branched-chain keto acid dehydrogenase kinase inhibitor. Michelle Lai here at Beth Israel, who's provided us liver biopsy samples, and many additional collaborators, including Nicola McCowan and Danielle Haslam, through which we've tried to study some of these mechanistic ideas in human populations through genetic epidemiological approaches, which I didn't really talk about today, and others involved in this process. So, and of course, our funders. So thank you, and I'd be happy to take any questions in the last 2.5 minutes. Yeah, so the question was, would CHRB in the adipose tissue have similar effects? And the simple answer is probably no. And some of that data is published when I was a postdoc in Barbara Kahn's lab. In adipose tissue, increased CHRB activity or increased de novo lipogenesis in adipose tissue seems to be associated with improved insulin sensitivity. And it may be that utilization of glucose to make lipid in the fat is okay, but utilization of glucose to make lipid in the liver is not. It also could very well be that CHB in the adipose tissue does other things that are protective, whereas CHB does distinct things in the liver which are not. So we know that the transcriptional targets, there are many that are conserved, but there are many that are non-overlapping between those two tissues. So CHB targets in the liver and adipose are not identical. So I think it's a really good question. Studies I would love to do, so let me know. But so what I would say is there is one good study looking at liver biopsies, gene expression liver biopsies from adolescents with fatty liver disease, from Sonia Caprio at Yale. And she showed clearly that in patients with fatty liver disease, young people with fatty liver disease had increased CHB beta activity in their livers. So it would suggest that this is a mechanism that's operative in that population, but I don't think anything's been looked at beyond that. Yeah. So the best way to do it is by mass spec assays. There are a number of platforms out there now that have good targeted metabolomic assays that include branched-chain amino acids, because this has become such a good marker. So you just have to work with one of those labs that's doing it. Yeah. Thank you.